I'm Ed Rybicki, and I'm going to be telling you about vaccines made in Africa for Africa at the University of Cape Town. Now, vaccine research at UCT spans a whole spectrum, as you can see from this line at the bottom here, from discovery through development, testing, upscaling through to clinical trial. These are not coordinated activities, but they could be. So in other words, one could go all the way from discovery through to clinical trial on the same set of vaccines that we would get via surveillance from departments of pathology and integrative biomedical sciences, through novel vaccine development and preclinical testing with the group's Biopharming Research Unit that I direct and the vaccine research group of Professor Annelies Williamson that are in the Institute of Infectious Disease and Molecular Medicine in faculties of Science and Health Sciences, through the UCT Animal Research Facility in Health Sciences, through production scale-up done at the Department of Chemical Engineering and the Faculty of Engineering and Built Environment, through to clinical trials, phases 1 to 3, done by the Faculty of Health Sciences. I note that people in health sciences have been responsible for COVID vaccine trials, tuberculosis vaccine trials, and HIV vaccine trials in recent years. However, I'm not going to be talking about the spectrum. I'm going to be talking about this in particular, the novel vaccine development and preclinical testing done by the BRU and BRG. This involves vaccine design, production and testing after identification of suitable candidates, investigation of novel production systems and particular vectors, novel vaccines, viral and bacterial candidate vaccines, developing small animal models for testing the vaccines, and then a lot of immunology done with the UCT Research Animal Facility, where one can do the small animal testing. There's also a BSL-3 animal facility for challenge models, and the potential of using knockout mice for studying particular immune responses in detail. Outputs are, and a neglected output is reagents for diagnostics. If you're working on novel vaccines, you're going to need novel reagents in order to assay for responses against them. Then the candidate vaccines for veterinary and human use, and then protocols and processes for testing and manufacture that could be handed on to, for example, partners in chemical engineering. Now, the range of candidate vaccines, to give you an idea of the kind of scope at UCT, just the BRU and BRG have been working on HIV-1 and SARS-CoV-2 in particular in the recent past. BRG is working on poxvirus-based veterinary vaccines for a variety of diseases using lumpy skin disease virus poxvirus. Then subunit and plant-based manufacture in the BRU involves various papillomaviruses, human and bovine, porcine and parrot circoviruses, African horse sickness and blue tongue orbiviruses as virus-like particles, influenza virus, hemagglutinin and DNA, various hemorrhagic fever virus glycoproteins in the study of emerging viruses, therapeutic HPV vaccines, and we're looking into brucellosis vaccines as well. I note that all the red asterisks denote vaccines that have been tested in the target species, which for DNA and poxvirus versions of HIV include humans. This activity doesn't only give rise to vaccines, of course, it gives rise to patents. We have a large list of granted and applied for patents, which include a complex patent family for papillomaviruses, for DNA expression vectors for cells, animals, and in fact plants, for plant-made influenza virus, hemagglutinin, orbivirus, virus-like particles, poxvirus expression-related patents, patents on HIV-1 envelope and SARS-CoV-2 S protein production in plants, a suite of patent applications on engineering virus glycoprotein glycosylation in plants, porcine circovirus, rotavirus, HPV therapeutic vaccines in plants. This is the largest vaccine-related patent portfolio from Africa. To give you a little bit of background on HIV, we were part of the South African AIDS Vaccine Initiative HIV Vaccine Development Program from 2001 to 2009. These put locally developed vaccines, locally designed and developed vaccines into clinical trials. They're the only set of HIV vaccines from Africa to make it into clinical trials, not once, but twice. These were vaccines based on 2 plasmids and modified vaccinia ankara, all of them expressing an envelope glycoprotein and a gag-related polyprotein. These went into these sets of clinical trials with the HVTN and SATVI that was published in 2016. They were perfectly good vaccines. They ended up being used as primers for boost with protein and looked to perform about as well as any other vaccines in development around the same time. However, the Strategic Health Innovation Partnership Program of the SAMRC started another HIV vaccine funding program in 2014, and we put in for next generation HIV vaccines because we knew that we could improve what we already had. And we improved it by making a mosaic protein for GAG, which is much more immunogenic than the native form, improved envelope designs, improved DNA expression vector to get much higher protein expression, and making a soluble protein subunit, which we hadn't done before, made in both mammalian cells and in plants. Now, this whole combination is much more immunogenic in preclinical studies than the previous versions. In fact, in head-to-head comparisons, it works much better. And we are hoping to get this into human clinical trial in the not too distant future. But now I give you, you've heard of Deep Impact, the movie, I give you Deep Impact, the virus. This is what SARS-CoV-2 has done to humanity and to our planet in the last 2 years. The moment we heard about it, Manny Margolin in our group designed a gene to be expressed in plants and mammalian cells. We've gone on to make that protein in plants. We've put genes into DNA vector and poxvirus-based vaccine vector. 3 vaccines, that's the subunit from plants, DNA, and poxvirus, have been tested in mice. They give excellent T-cell responses and neutralizing antibodies. The T-cell responses, especially nice to have for the protein-based vaccine because we didn't expect it. These are going to go into hamsters for SARS-CoV-2 challenge in the UCT BSL-3 unit. The challenge model is going to be established in collaboration with the ICGB in Cape Town and the universities of Stellenbosch and Western Cape to test vaccine efficacy. Now, I will point out that this entire program, starting February last year, the only funding we've had for it is from UCT. None of our national funding agencies have seen fit to fund us for it, but yet we may still be able to make vaccine or vaccines. Thank you.
Hello, my name is Thanos Kotsiopoulos, and today with my colleague Siew Tai, we're here to present the role of bioprocess engineering in the vaccine sector. At the Center for Bioprocess Engineering Research, we design and model process frameworks that accelerate the development of bioprocesses to optimize the production of vaccines with minimum risk while still remaining cost-effective. This is important in the context of developing economies such as South Africa, where vaccines need to be cost-effective to be available for all members of the population. So, as we know, in the development and discovery of vaccines, scientists are involved in selecting a suitable vaccine type for the particular disease they are fighting. With recombinant technology and more recently mRNA vaccines, newer vaccines are being developed using different cell lines, such as bacteria, yeast, and plants for their production. At CeBER, we have worked with several cell types from bacteria to fungus to plants and mammalian cells, and we have the skills and knowledge in growing these various cells efficiently. Here we have considerable experience in selecting appropriate media type and optimizing conditions to enhance growth for maximum expression of the target proteins for vaccine production. However, in using different cell types to produce vaccines comes challenges in growing them optimally for products. Most of these cells have an operating window in which they grow. If these conditions are not maintained, we can have poor growth, low yields, and productivities. And even at times, the efficacy of the vaccines themselves can be compromised. As bioprocess engineers, we have the expertise in avoiding these types of inefficiencies. For example, in the development of animal and human vaccines, ensuring adequate mass transfer is shown to be very frequently to be contingent on the reactor configuration that can deliver adequate mixing for oxygenation of the system, which ultimately leads to the cost of production. So given that knowledge that there are external factors that need to be dealt with when developing a vaccine, scaling up the process needs to be done methodically to ensure reproducibility at various scales. At CIDER, we have various platforms ranging from the milliliter scale up to 1 liter reactors and increase all the way up to 70 liter bioreactors, which allows us to bring the vaccine development from the bench to the pilot scale. We also have the capabilities to design novel, low energy, low shear bioreactors. For example, the horizontal tubular bioreactor, where we have successfully grown mammalian cells in suspension up to 3 liters. But the question arises to how do we successfully scale these processes from discovery in the lab to pilot or commercial scale? Several scale-up parameters need to be considered and tested, which may hamper production volumes and rates, and need to be optimized at various stages before the technology is commercialized. Questions on how may we move from batch or semi-batch operation to continuous processes are aspects that we investigate to maximize product capacities with minimum delay in the production line. Although the bioreactor may be considered the heart of the production process, we consider the full chain of technology when developing vaccines. After production of the bioactive, these products need to be isolated and purified. This goes through various stages where we choose the right technology to achieve the desired purity while keeping costs at a minimum. At CeBER, we have various platforms to do this, ranging from cell separation using centrifuges to filtration systems or separation by solubility and selective absorption. We also have a means of isolating and purifying the heat-sensitive bioactive materials by precipitation and chromatography techniques, a major cost when high purities are needed in the process. At CIDER, we also have skills outside of the laboratory, where we develop process flow sheets for vaccine manufacture. This exercise is important for the vaccine industry in South Africa, as the requirements and cost structures are different to developed economies such as those in Europe and America. We have done several of these comparative studies for various biopharmaceutical industries, where we have used these case studies to find the most cost-effective technology platform to make the vaccines via a comprehensive techno-economic study. It's an important exercise that should be done at the initial stages of the projects, as together with techno-economic feasibility studies, any cost-sensitive areas or bottlenecks in the production process may be identified that may need to be revised at bench scale. We use this approach to determine the efficacy of scaled operations and its subsequent translation to commercial scale. This analysis allows us to consider flexibility in reagents. For example, substrates used in the laboratory for synthesis may have an impact on the cost and viability of the process. We also look at whether stainless steel reactors, which need continual cleaning place strategies to maintain sterile environments, can be replaced by single-use reactors or even hybrid systems to lower overall operating costs. In each case, the impact on performance or yield based on criteria are evaluated and revised based on scaled outcomes. This informs the potential production routes that may be used to standardize the manufacturing process for a range of vaccine candidates. We also are skilled in building vaccine manufacturing plants which require specialty skills in clean room design and technology. As we can see from the flow sheeting exercise, it is necessary to design these processes to meet the regulatory requirements from the beginning. It is for this reason that bioprocess engineers should be involved in the development of vaccines from the start.
Hello, my name is Tom Scriba, and I want to tell you more about the South African TB Vaccine Initiative, or SATVI. We are a vision-driven research group at the University of Cape Town, and our primary focus is tuberculosis. Why? Because it has killed the most people on Earth of any infectious disease, and because it's clearly an enormous global public health problem, and especially in Africa, in South Africa, and other areas of the world. The graph on the right shows the number of TB cases in millions per year over the last 20 years, and you can see that the trajectory is coming down, which is good, but unfortunately it's coming down too slowly, and in fact we missed the 2020 milestone of the WHO and Stop TB partnership. And also there's actually been a slight upturn in the last year or two, probably caused by the COVID-19, or certainly contributed to by the COVID-19 pandemic. So clearly an important problem to work on, and one that affects us directly here in Africa and in South Africa. Who are we? We're a very large research group. The picture at the bottom shows our staff and students just before COVID-19. We are comprised of doctors, nurses, clinical field workers, many others, including a laboratory team, students and postdocs. And we work in communities in and around Worcester, which is about 110 kilometers from Cape Town. And that is where most of our staff members are based, and where TB is particularly a large problem. We work highly collaboratively with many around the world and span questions that we can address all the way from clinical vaccine development through to basic immunology questions in humans. So how do we do what we do? Well, our primary goal is to develop TB vaccines. And over the last 20 years that we've been around, we've completed 29 TB vaccine trials of 9 different vaccine candidates, as well as the TB vaccine that's already been in existence for 100 years, BCG. So clearly a lot of work has been ongoing, and we have a lot of experience in vaccine development. We then couple this with TB immunology. And so we collect specimens in humans that take part in our studies and participants. And I just want to show you one example here of the type of immunology study that we've done to try and understand the pathogenesis of TB. So one of the things that's particularly difficult in TB is the slow progression that we see in humans after they become infected. And in fact, many people do not progress to active TB disease. But in those who do, there's a slow progression that occurs from what we call latent infection, asymptomatic. We can't really find the bacteria in these people, through subclinical phases where people do not have symptoms, but they have ongoing disease processes, and then all the way through active TB disease where we can find the bacteria in sputum. And so on the right, you can see these are the results of a blood gene expression test that we've developed, which allows us to track the type of inflammatory response that's ongoing. This is over a 2-year period in red. In the red bars, you can see these are people who progress from TB infection all the way through to active TB disease, which would be the time point 0. I'm showing this in months over here. You can see that this inflammatory response develops and gets much higher all the way to the top there. And that's when we can diagnose TB. And then in people who go on to TB treatment, that inflammatory response reverses, and we can detect that as well. And so this kind of a tool can be used to identify people who are on their way to progressing to TB and would benefit from early interventions, such as antibiotic treatment, before they actually have symptoms and have active disease. And in the same way, we can also track how well people are responding to the antibiotic treatment. Of course, TB vaccine development is our big focus. And I just want to mention one example, because this is really a very exciting moment in TB vaccine development, because this one product called M72, which is just 2 TB proteins of 4,000 in the bacterium that have been formulated in an adjuvant by GSK. And this adjuvant is actually partially made from a natural product, from a tree. It's called QS21. And so this vaccine was recently tested in a very large study of 3,500 odd adults who already have evidence of infection with tuberculosis, but are not yet sick. These were then randomized to receive the vaccine or placebo. And over a 36-month follow-up period, we looked for the endpoint of TB disease. And so we were testing whether this vaccine can prevent progression to active TB disease in people who already have evidence of infection with the bacterium. And so the exciting result that we've been waiting for many years is that we saw some efficacy at 50% against TB disease. And so you can see that these curves diverge. What we're looking at is when the curve drops, we are seeing people who have active TB, and you can see that in the placebo group in blue, that line drops, or the graph drops lower than it does in the M72 group, clearly showing evidence for protection. Very exciting. Not yet sufficient. The sample size in the study was not large enough to allow licensure of the vaccine. But what it does allow us to do is to look at, to understand what is protecting. And so, of course, in the laboratory, we measure the immune response to vaccination, and this slide shows that we induce a very nice T-cell response by this vaccine. But we don't know in a small study like this whether this particular immune response is responsible for that protective effect. And so another thing that we are currently spearheading, Elisa Nemes and myself are part of, are co-PIs of this program, and which is led by Nicole Fromm from the Gates Medical Research Institute. And this is specifically to identify immune correlates of protection using samples collected from those that are protected and those that are not protected from this particular trial that I'm telling you about, or that I told you about. This is a very large effort. We solicited input from around the globe, and 17 different assays and approaches have been selected. It's funded by the Gates Foundation, the Gates MRI, and the NIH of the US, and 12 different laboratories will be taking part. And this is clearly very exciting because we're going to cover the entire spectrum of immune responses that we think are possibly involved in this. So I'll end there, and thank you for your attention.
Hello, friends and colleagues. I am grateful to Sue Harrison for the opportunity to share some of our departmental contributions to COVID-19 vaccine scholarship. I apologize if I look a little bit casual. This is the fanciest shirt that I brought along on holiday. The Department of Medicine is a large and complex department which plays a leading role in medical education, research and scholarship, and provides clinical services to communities in the Western Cape. And in the case of our highly specialized services, to patients throughout Southern Africa. Of course, we relish the opportunity to be able to contribute to COVID-19 scholarship in the last 2 years, work which has been both impactful as prolific. I will share some of these contributions to COVID-19 vaccine research from 4 groups within our department, but I wish to acknowledge the multiple efforts of many, many groups within our department answering important and fundamental questions on COVID-19 in our context. The first group I wish to review work on vaccines from is the Desmond Tutu HIV Center and Foundation, which is led by Linda Gale Beckham and her many colleagues and contributors. The first important contribution was, of course, the completion of the Sisonke trial, which was a large implementation open label trial of the single dose Johnson & Johnson vaccine given to healthcare workers in this country, critically at a time when vaccines were not yet available in South Africa. And in this valiant effort, managed to vaccinate nearly 500,000 healthcare workers. In addition to this important Sisonke effort, Linda Gale and her colleagues have also contributed to the Ensemble trial, which was a trial assessing the single-dose Johnson & Johnson vaccine in prevention of COVID-19 in adults. This work has now been completed. They've also contributed to the Ubuntu trial, which is a trial testing a 3 rather than 2-dose strategy for the Moderna mRNA vaccine. This is planned to be completed in October of 2021. They also led locally the implementation of the Crown Coronation trial, led jointly by Linda Gale Becker and Bruce Picard, testing the efficacy of the MMR vaccine against COVID-19. In addition, a sub-study of the Sisonke trial looked at both B and T cell responses in about 600 individuals of different ages, co-infected with HIV and with comorbidities. They've also looked at one of their sites at Masiphumelele on an open label trial of a half-dose Johnson & Johnson vaccine with a Pfizer booster. And this is work that is currently ongoing. As part of the co-VPN consortium, they've also contributed to the design of a number of studies that include immunologic, virologic, clinical data for future COVID-19 vaccine efficacy trials, and these are currently ongoing. And other than the work that they've done in leading scholarship on vaccines, colleagues at the Desmond Tutu HIV Center and Foundation have also contributed to the vaccination of nearly 50,000 healthcare workers at their sites. And these constitute about half of the healthcare workers vaccinated in the Western Cape. The CIDER Africa group, led by Graham Menkes, has also made substantial contributions in vaccine work. They also contributed to the running of the Ensemble trial at Khayelitsha. This was led by Amy Ward. And in addition, also contributed to the coordination of the Sisonke trial at Khayelitsha. They have contributed to the ProViva SA study, which is a phase 1 trial of Immunity Bio's vaccine in South Africa. And they've contributed 41 patients to this trial. In addition, they've also contributed to phase 1 and 2 trials of a booster trial of the H85 vaccine in those who've previously received the single-dose Johnson & Johnson vaccine. And this is work that is also being conducted by the Desmond Tutu HIV Center at their Emavundleni site. The Center for Lung Infection and Immunity, led by Keaton Data, has also contributed substantially to vaccine work. They contributed towards the phase 1 and phase 2 of the ChAdOx trial in this country, as well as the phase 2A and B of the Novavax trial. And you can see the publication from that work, as well as the Providence trial, which tested the safety, tolerability, and immunogenicity of a novel Pro-CL-002 vaccine against the Pfizer vaccine. And the publication is included in this slide. Our group, together with Wendy Beggars and others based at the IDM, as well as Penny Moore at the NICD, looked at immune responses of healthcare workers to the single-dose Johnson & Johnson vaccine. and demonstrated that infection prior to vaccination significantly boosted cross-reactive antibodies and their cytotoxicity, binding, and neutralization, as well as moderately boosted T-cell responses. Further, we showed that infecting virus spike sequence determines the cross-reactivity of neutralizing responses, and this has got huge implications for second-generation vaccine design. So I wish to conclude by acknowledging again the important work that is being done by many groups within our department. I've shown you the multiple COVID-19 research studies ongoing in our department, and these span from basic sciences to the clinical sciences, epidemiological and surveillance studies, multiple randomized controlled trials, and some population health studies. The department and many PIs within it has made significant contributions to SARS-CoV-2 vaccine research. And many of the vaccine studies I mentioned are currently ongoing. And I think what has been a wonderful feature of this work has been the benefit from resident expertise within our own department, within the IDM, and a number of other institutes that we work closely with, as well as our many other collaborators within and outside of UCT. The presence of infrastructure that has enabled the conduct of this work. And of course, one cannot downplay the importance of the strong collaborations between us and many partners that have enabled this work. Thank you very much for your attention.
Good evening. I'm going to be talking about the Western Cape Provincial Health Data Centre, which has been developed as a partnership between Western Cape Health and UCT, and its role as a resource for understanding disease epidemiology and evaluating vaccines. My talk will be divided into two parts. First, I'm going to explain how the Provincial Health Data Centre works. And secondly, I'm going to highlight some of the uses of the PHDC. So first, how does it work? The Western Cape has a population of about 7 million people, of whom about 75% use the public sector for health services. When these people attend a public sector facility, a folder is either retrieved or created, and what's central to the PHDC is that the same folder number or unique identifier is used for all health services that are delivered. This allows us to merge data from a range of different service delivery systems. And although most data is limited to the public sector, because COVID is a notifiable medical condition, we have also been the provincial system for reporting on all COVID-19 diagnostic tests and vaccines. Importantly, the data is derived directly from the patient's interaction and receiving a health service, such as a laboratory test or a drug being dispensed. So there's no additional data capture or parallel data capture system. The system also differs from health insurance claims, which rely on disease coding by the provider. Instead, through a process that we call data beneficiation, what we do is to use the actual patient healthcare data to infer a range of different conditions. So for example, if a patient has a positive HIV diagnostic test, or if they've had an HIV viral load or a CD4 test, or if they're on antiretroviral therapy, we can reasonably infer that they have HIV. Here you can see just some of the conditions that we're able to infer through data beneficiation. And so you can see, for example, that we have about 400,000 people living with HIV known to the province and about 100,000 new pregnancies per year. You can imagine that this data could be used to identify groups of patients who might be suitable for a particular vaccine trial. We can also identify outcomes that might be important for a vaccine trial, like an infectious disease outcome, and in particular, an outcome that could happen long after the trial is finished, like cervical cancer for HPV vaccine. And we could also look at adverse events. The beneficiated data is used primarily to improve patient care by effectively creating a virtual electronic medical record, which can then improve continuity of care, and also by generating reports at facility or program level in order to monitor and improve service delivery. But a spin-off benefit, which is what I'm going to focus on today, is that the data can be used to help us understand disease epidemiology and assess the effect of interventions. And of course, for today, we're going to focus on vaccines. So what are some of the uses of the PHDC? Well, as soon as you have this kind of wealth of data, everybody wants a dashboard, and COVID really highlighted dashboards for us. And so since the start of the epidemic in the Western Cape, we've had an interactive public-facing dashboard, which allows the public to drill down into our COVID data and has had over 3 million views. You can see very nicely in the graph at the bottom that the number of daily dashboard views has mirrored the waves of the epidemic. But COVID isn't our only disease, and so we also have recently launched a TB dashboard, which also has a dark version shown on the right, focusing on drug-resistant TB. What about going beyond just tracking infectious diseases to actually understanding their epidemiology? Well, even before the peak of the first wave of the COVID pandemic in the Western Cape, we were able to use our data on about 3.5 million active adult public sector patients to show that when we adjusted for all the risk factors for COVID death that had been shown from other countries, both HIV and TB were associated with about a 2 to 3-fold increased risk of death. We were the first study to show this, and the results were subsequently confirmed in studies from the US and the UK. We're currently using our granular data on comorbidities to identify patients at high risk of COVID death on account of their age or comorbidities who have not yet been vaccinated, so that when they come for their next clinic visit or drug pickup, they can be vaccinated as they visit the health service. But perhaps of most relevance to this webinar is the ability of PHDC data to assess the real-world effectiveness of vaccines. And this was very nicely illustrated in our analysis of the AD26 or J&J COVID vaccine, which was administered to healthcare workers as part of the phase 3B Sisonke trial. The challenge with assessing this kind of non-randomized study is that those who choose to be vaccinated, and especially those who access the vaccine early, may be very different from those who don't get vaccinated or get vaccinated late. And in addition, in the background, the force of infection was changing as the vaccine was rolled out. So we used 3 different analytic approaches to address this challenge in analyzing the data of nearly 35,000 Western Cape healthcare workers who are employed by the Western Cape Department of Health, of whom about 65% were vaccinated through Sisonke. The Kaplan-Meier shows the results of one of these methods, and reassuringly, all 3 approaches yielded similar results with a vaccine efficacy of around 75% against hospital admission and 80% against death. It's also important to be able to assess the clinical severity associated with different waves, which have been driven by different variants, and to see whether during the 3rd wave, which was driven by Delta, vaccines protected against severe outcomes in the context of Delta infection. Here, what you can see is that after adjusting for all the normal risk factors associated with COVID death, the 3rd wave was associated with about a 9% higher risk of death compared to the reference or 2nd wave, which was driven by Beta, which in turn was more severe than the 1st wave. But these results are not adjusted for the fact that during the 3rd wave, at least some people were vaccinated. Once we take vaccines into account, we see that for the unvaccinated, the 3rd wave was actually 26% more severe than the 2nd wave, and that both partial and full vaccination provides strong protection against severe COVID or COVID death with a nice dose response effect and the fully vaccinated being more than 4 times less likely to die from COVID compared to the unvaccinated. In summary, the Western Cape Provincial Health Data Centre is a valuable resource to help us understand our disease epidemiology, to identify candidates and communities for vaccine trials and provide data enhancement, such as outcomes and adverse events, to guide and optimize the implementation of vaccines, and to assess vaccine efficacy and safety, not just of COVID vaccines, but also of other vaccines such as childhood vaccines and vaccines that may be introduced in the future, such as against Group B strep. I end by acknowledging the partnership between the Department of Health and UCT that's made the PHDC possible, the hard work of the core team, and support from multiple technical partners and funders.
Thank you for this opportunity to give a glimpse into the work of the Vaccines for Africa initiative in the vaccine research space in the context of Africa within the University of Cape Town. VACFA fills a niche that I believe demonstrates what the American educator Ernest Boyer would call the scholarship of engagement. The journey of a vaccine starts in the preclinical space, usually in the laboratory. From here, the promising candidate will move through various stages to hopefully emerge after undergoing clinical trials declared both safe and effective and getting registered. It will then need to jump the gap from the research space into daily clinical use. While the first part is carried out by scientists, the responsibility of the second part falls on the shoulders of policymakers and implementers of healthcare programs. In practice, the choice of a vaccine and how to best deploy it in the public space is informed by a variety of reasons, such as specific epidemiological considerations of both population and disease, the cost effectiveness of the vaccine, acceptance, etc. How is the policy maker to make a decision that is both correct and contextual to his or her jurisdiction? It becomes immediately clear that there's a chasm between science and policy that needs to be bridged. The Vaccines for Africa initiative was created specifically to bridge this chasm. This academic group was founded in 2009 under the leadership of Emeritus Professor Gregory Hussey. It is located in the Division of Epidemiology and Biostatistics in the School of Public Health and Family Medicine within the Faculty of Health Sciences of the University of Cape Town. As VACFA endeavored to bridge this gap between science and policy, it has done so by demonstrating a scholarship of engagement that has come in the form of promoting awareness of benefits of vaccines within the African continent, generating programs for building capacity for Africa. This has demanded that VACFA, at its core, engages in research on vaccines and immunization practice that generates evidence for informed decision-making. This has finally culminated now in our current flagship pioneering project, which I will mention shortly, the Vaccinology Regional Scientific Hub for evidence-informed decision-making in Africa. As I mentioned, the generation of evidence on which to base decisions is at the core of the work of VACFA. As a result, VACFA spends a lot of time doing systematic reviews on different vaccinology-related topics. This includes research on evidence to support clear quantification of the burden of disease due to vaccine-preventable infections in different population groups, effectiveness of different vaccination strategies, reviews of health systems for supporting immunization programs. It involved systematic review on trade-offs between adverse events and different vaccines, cost effectiveness of vaccination strategies, etc. Over the last decade and a half, in addition to other vaccine-related research output, VACFA members have published thousands of peer-reviewed systematic reviews. Secondly, the need for evidence-based vaccinology has demanded that VACFA gets directly involved in supporting policy advices to the Ministry of Health of South Africa. VACFA members, including our postgraduate students, both past and current, take part in supporting working groups that advise the Ministry of Health with respect to vaccination practice. By the very nature of its role, VACFA is to be a melting pot of academic disciplines, such as public health, epidemiology, statistics and mathematical modeling, health economics, immunology, clinical virology and microbiology, clinical practice disciplines, health systems, design thinking, etc. In addition to working with various academic disciplines to reach across the gap, VACFA works with organizations and government officials involved in immunization programs, as well as with key industrial players. This multidisciplinary engagement goes beyond the borders of South Africa. An example of this is the design thinking workshop on maternal immunization that was organized here in Cape Town, that VACFA was central to in 2019. This involved the D-School and WHO, and it had participants from 6 African countries, including Benin, Ethiopia, Gabon, Senegal, South Africa, and Tanzania. An important role of VACFA is to share information on vaccination and immunization practice beyond publications of peer-reviewed manuscripts. VACFA hosts postgraduate students, master's, doctoral, and postdoctoral students with the express goal of training them and teaching them in evidence-based research in vaccinology, largely on topics that are biased towards vaccinology needs of Africa and other low and middle-income countries. Another important platform has been the hosting of the annual African Vaccinology course. The course provides participants with essential and up-to-date knowledge on human vaccines. Participants get to discuss the successes, challenges, and opportunities to improve national immunization programs in Africa. In addition, the course provides a forum for members of NITEX to share national immunization issues and identify areas for collaboration and technical capacity development, fostering communication and networking among African vaccinologists. Over the last 15 years or so, the annual African Vaccinology course has trained more than probably close to 1,000 participants, covering most of the African continent. And you can probably see also how wide the backgrounds of the faculty is that take part in this course. And that composition, again, demonstrates a level of integration required to bridge the gap between science and policy. In 2017, a program funded by the Bill and Melinda Gates Foundation for supporting independent immunization and vaccine advisory committees called CIDER ended in response to the efforts of WHO Africa region to fill the service gap left behind by CIDER. The VACFA team reached out with a proposal. Extensive engagements took place between the University of Cape Town through VACFA and WHO. Both the region for Africa here at home in Congo-Brazzaville, as well as a NITEC support center at WHO HQ in Geneva. This collaborative effort involved representatives of national immunization advisory committees from multiple countries in the African continent and included a scoping exercise that ultimately led to the proposal of forming a regional scientific hub for evidence-informed decision-making in vaccinology for Africa. This project is being led by my colleague, Dr. Benjamin Kagina. This project, which is funded for the next 5 years, is only possible because of the generous support of the Wellcome Trust. The role of the hub will be to provide evidence-informed decision-making workshops, running vaccinology courses, producing training materials for vaccination practice. The hub will develop manuals covering generic and specific issues in vaccinology that affect the African continent. All these activities are geared towards closing that gap between science and policy, allowing the African policymakers to make defensible decisions standing on solid scientific ground. Thank you.